A multi-state salmonella outbreak is linked to onions imported from Mexico. According to the CDC, the produce sickened more than 650 people in 37 states. Right now, no deaths are reported, but nearly 130 people went to the hospital. According to the CDC, the outbreak is linked to red, yellow, and white onions distributed by ProSource Inc. and imported from Chihuahua, Mexico. Investigators are still working to see if the outbreak is linked to other onions or suppliers. Katie Johnston for CBSN, Los Angeles.
When designing a lamp experiment, 4 to 6 primers are required, specific to identified regions of target DNA or RNA sequence. Amplification initiates from strand invasion by one of the inner primers, and a strand-displacing DNA polymerase extends the primer and separates the target DNA duplex. The first product is then displaced by synthesis, initiating from an outer primer which anneals to an upstream target region. As it is displaced, the end of the product forms a self-hybridizing loop structure due to inclusion of a reverse complementary sequence in the inner primer sequence. This annealing and displacement cycle repeats on the opposite end of the target sequence, and the resulting product is a short dumbbell structure that forms the seed for exponential lamp amplification. This lamp dumbbell structure contains multiple sites for initiation of synthesis, from the 3' ends of the open loops and annealing sites for both the inner and loop primers. As amplification proceeds from these multiple sites, the products grow and form long concatenamers, each with more sites for initiation. The result is a rapid accumulation of double-stranded DNA and amplification byproducts that can be detected by a variety of methods.
That is, typhoid and paratyphoid fever, caused by Salmonella typhi and Salmonella paratyphi A and B respectively. Antibodies specific to flagellar antigen H and somatic antigen O of Salmonella species usually become detectable in blood 7 days after the onset of infection in individuals infected with typhoid bacilli. This test facilitates quantitative estimation of antibodies to Salmonella antigens in human serum by slide and tube agglutination test. Principle: when the colored, smooth, attenuated antigen suspensions are mixed and incubated with patient serum, anti-Salmonella antibodies present in the patient's serum react with the antigen suspensions to give agglutination. Agglutination is a positive test result indicating presence of anti-Salmonella antibodies in the patient's serum. Negative test result is indicated by the absence of agglutination. Primary sample: no special preparation of the patient is required prior to sample collection by approved techniques. Collect 2 milliliters of venous blood in a plain red-topped vacutainer. Do not use hemolyzed and turbid samples. Though freshly collected serum is preferable, samples can be stored at 2 to 8 degrees centigrade for up to 72 hours in case of delay in testing. Reagents and consumables: Salmonella typhi O antigen suspension, Salmonella typhi H antigen suspension, Salmonella paratyphi AH antigen suspension, Salmonella paratyphi BH antigen suspension. Polyspecific positive control: glass slide with 6 reaction circles, applicator sticks. Equipment: incubator, timer, 0.1 milliliters and 1 milliliter serological pipettes and droppers, 36 Vidal tubes or Kahn tubes for each sample. Physiological saline, test tube rack. Test procedure: bring reagents and samples to room temperature before testing. Shake and mix antigens well before dispensing. Slide screen method: place 1 drop of positive control onto a reaction circle of the glass slide. Place 50 microliters of physiological saline onto the next reaction circle of the glass slide. Place 1 drop of patient serum to be tested onto each of the required number of reaction circles. Add 1 drop of O antigen suspension to the reaction circles containing positive control and physiological saline. Add 1 drop each of the antigen suspensions, that is O, H, AH and BH, to the reaction circles containing the patient's serum. Mix contents of each circle uniformly over the entire circle with separate mixing sticks. Rock the slide gently back and forth and observe for agglutination macroscopically at 1 minute. Interpretation of results: observe for agglutination or formation of clumps in the wells. Compare it with the positive control. Agglutination is a positive test result and indicates presence of the corresponding antibody in the patient's serum. Absence of agglutination is a negative test result and indicates that the corresponding antibody is not present in the patient's serum. As the test wells show, agglutination with O and AH antigens, the corresponding antibodies, namely Salmonella paratyphi O and H antibodies, are present in the patient's serum. Hence, the infecting organism is Salmonella paratyphi A. Slide semi-quantitative method. This method is recommended for obtaining quick approximate titers only for the antigens that tested positive in the slide screening test. Using a pipette, place 80 microliters, 40 microliters, 20 microliters, 10 microliter, and 5 microliter of patient serum to be tested on 5 different reaction circles on the glass slide. The corresponding dilutions obtained will be 1 in 20, 1 in 40, 1 in 80, 1 in 160, and 1 in 320 respectively. Add to each reaction circle a drop of the antigen which showed agglutination with the test sample in the screening method. In our demonstration, it will be O and AH antigens. Using separate mixing sticks, Mix the contents of each circle uniformly over the reaction circles. Rock the slide gently back and forth. Observe for agglutination macroscopically within 1 minute. Interpretation of result. Agglutination is a positive test result. The titer is reported as a reciprocal of the highest dilution, which shows a positive test result. Quantitative tube method. Take appropriate number of sets, one set for each antigen suspension of 8 count tubes or test tubes, and label them 1 to 8. Pipette 1.9 milliliters of physiological saline into tube number 1 of all sets. To each of the remaining tubes, that is 2 to 8, add 1 milliliters of physiological saline. To tube number 1 of all sets, add 0.1 milliliters of serum sample to be tested and mix well. Transfer 1 milliliters of the diluted serum sample from tube number 1 to tube number 2 and mix well. Transfer 1 milliliter of the dilute serum sample from tube number 2 to tube number 3 and mix well. Continue the serial dilution till tube number 7 in each set. Discard 1 milliliter of the diluted serum from tube number 7 of each set. Now, the dilutions of the serum sample achieved from tube number 1 to 7 respectively in each set is as follows: 1:10, 1:20, 1:40, 1:80, 1:160, 1:320, 1:640. Tube number 8 in all the sets serves as a saline control. To all the tubes, that is 1 to 8 of each set, add 1 drop of the respective well-mixed antigen suspensions from the reagent vials and mix well. Cover and incubate overnight at 37 degrees centigrade. After incubation, dislodge the sedimented button gently and observe for agglutination. Interpretation of result. The tube test measures the titer of antibodies present in the patient's serum. Titer is defined as the concentration of an antibody determined by finding the highest dilution of serum at which it is still able to cause agglutination of the antigen.
QuickView Salmonella typhi antigen test is an in vitro qualitative immunochromatographic assay for the rapid detection of Salmonella typhi antigens in human stool or serum specimen. The test results are intended to help in the diagnosis of Salmonella typhi infection and to monitor the effectiveness of therapeutic treatment. This kit provided by Lumicruck Diagnostics contains 25 test devices and 2 sample buffer droppers. Each pouch contains a transfer pipette. To use the test, bring the kit components to room temperature. Add 12 droplets approximate 500 microliter of sample buffer from provided dropper. Pick approximate a quarter gram of collected stool and mix it well with sample buffer in the reaction tube. Allow the mixture to sit for 5 minutes so to allow the large particles to settle. Place the device. At 5 minutes, use the provided pipette to transfer a sample from the upper layer of the stool extract. Add 3 drops to the sample well. Start the timer. Read the result at 20 minutes. Typhoid fever is a life-threatening illness caused by the bacterium Salmonella typhi, and was observed by birth in the mesenteric nodes and spleen of fatal cases of typhoid fever. The infection is acquired typically by ingestion. On reaching the gut, the bacilli attach themselves to the epithelial cells of the intestinal villi and penetrate to the lamina and submucosa. They are then phagocytosed, thereby polymorphs and macrophages. The ability to resist intracellular killing and to multiply within these cells is a measure of their virulence. They enter the mesenteric lymph nodes, where they multiply and, via the thoracic duct, enter the bloodstream. A transient bacteremia follows, during which the bacilli are seated in the liver, gallbladder, spleen, bone marrow, lymph nodes and kidneys, where further multiplication takes place. Towards the end of the incubation period, there occurs a massive bacteremia from these sites, heralding the onset of the clinical symptoms. Read results. If you are interested in having business with us or interested in any of our products, please feel free to send your inquiries to info@lumicruck.com or visit our website at www.lumicruck.com.
Fast Track Diagnostics, syndromic real-time PCR, multiplexing that serves the clinical need. Let's see why this is important to you.
Imagine a scenario where a person falls ill. They go to the doctor for a diagnosis. The doctor is concerned about the patient's symptoms and overall presentation and decides that further investigation is required. A sample is taken, for example a blood sample, and a test is ordered from the lab based on what the doctor expects to be the most likely pathogen causing the illness. The test takes place, and the results come back negative. A second test must then be ordered. It also comes back negative. A third test is ordered, and the cycle repeats until a positive result is found. Time ticks by. This repeated testing is bad for the patient, frustrating, wastes time, reagents, and money, and the patient remains untreated as he waits for a diagnosis, possibly getting sicker. Worse yet, the patient could be prescribed unnecessary medication, like antibiotics for a viral illness, before the test results are confirmed.
Now imagine the same scenario, but the doctor can order a test from the lab for all probable pathogenic causes, viruses, bacteria, fungi, or parasite, at the same time using one test. This is syndromic real-time PCR multiplexing. Yes, it saves time, resources, reagents, and money. But most importantly, it is better for the patient because it is an effective aid to fast clinical diagnosis, reducing morbidity and the risk of mortality, particularly in cases involving immunocompromised patients. Wouldn't that be better?
At Fast Track Diagnostics, we manufacture the largest range of syndromically grouped real-time PCR tests available, covering all the major disease groups like respiratory infections, gastroenteritis, sexually transmitted infections, fever, and rash. We offer large and small panels of qualitative tests for screening and differentiation, many of which can be used in flexible combinations for cost-effective testing, and where an understanding of the viral load or level of infection is critical, we offer quantitative tests too. Our kits contain all the reagents required, including optimized primers and probes, pooled positive controls, extractable internal controls and negative controls, and Fast Track Master Mix, our one-step RT-qPCR enzyme.
Now let's look at the protocol. It is so simple, and the same for all of our kits. Step 1. Extraction is performed on the sample. Our kits work with a wide range of automated, semi-automated, and manual extraction methods. Step 2. PCR setup. Pipette the required volume of each reagent onto the plate. Now you're ready to begin the run. It's so easy. There's only one pipetting procedure and the same volume of reagents, no matter what thermocycler you use. And our assays are compatible with 10 of the most widely available, with more being evaluated. Also, because all of our kits have exactly the same protocols, in a single run, different kits, testing for a range of viruses, bacteria, fungi, or parasites, can be used at the same time on the same plate.
Then comes the most important part. With our real-time PCR kits, the doctor can have fast, comprehensive, accurate results within about 2.5 hours. Good news for the patient. All our customers can get expert support, scientist to scientist, direct from us, or from one of our international partners. And our kits are regularly evaluated using external quality control panels. They are subject to rigorous production checks and are CE labeled, so you can trust Fast Track Diagnostics kits to give
True positives,
true negatives.